Hi everyone, this is the final component of this topic on the digestive system, and it's on nutrition and metabolism. These are the learning objectives for this part of the topic, and they are to be able to define the terms nutrition, macro and micronutrient, and essential and non-essential nutrient. Be able to define what we mean by metabolism and outline the processes of anabolism and catabolism. Explain the role of ATP and the ATP-ADP system in metabolism. Be able to state the various dietary sources of carbohydrates, lipids, and proteins. Outline carbohydrate metabolism and the generation of ATP, including the role, very briefly and very broadly, the role of glycolysis, the citric acid cycle, and the electron transport chain. Explain what is meant by the terms anaerobic respiration and aerobic respiration, and then finally differentiate between glycogenolysis, and gluconeogenesis. So nutrition refers to all of the food or nutrients that we eat. And nutrition has been described by several different bodies, including the Council on Food and Nutrition of the American Medical Association, as kind of like the science of food. So, when we're talking about nutrition, we're talking about the types of food we eat and the way that these types of food interact with each other and their relation to our overall health. But also, how we ingest that food, how we digest it, how we absorb it, how we transport it, how our cells kind of utilize it, and how these nutrients are excreted from the body. Now, macronutrients are nutrients that the body needs in large amounts. So in terms of macromolecules, these are carbohydrates, lipids, and proteins. And in terms of macrominerals, so minerals that are needed in large quantities, this includes sodium, chloride, and calcium. And because we kind of need these in large amounts, they're often called bulk. Conversely, micronutrients then are nutrients only needed in small amounts. So these include the microminerals that I'm sure you've heard of them being called the trace elements, such as iron, iodine, and zinc. And of course, iron needed for transport of oxygen in red blood cells, iodine in particular being important for thyroid hormone production. Essential nutrients are nutrients that must be obtained from the body because the body can't actually make them. And non-essential nutrients are nutrients the body can synthesize if it is given the proper raw materials. So the body can assemble these non-essential nutrients if they have the essential nutrients as the building blocks. Now, we've got a few important definitions on this slide. The first one is looking at metabolism. And metabolism is the combination of all the chemical reactions occurring in the body. And this includes how the body utilizes nutrients. And there are two main processes. They are catabolism and anabolism. We'll define those now. Catabolism is the breakdown of complex molecules to form simpler ones. And in doing this, this releases energy, heat energy and chemical energy. And chemical energy must be transferred to ATP or adenosine triphosphate molecules, which then supplies energy directly to cellular reactions that require it, okay? Anabolism then is a process of synthesis, so things getting bigger. Think anabolic steroids and the effect they have on the body. So making big molecules out of smaller ones. For example, making proteins from much smaller single amino acids. And both catabolism and anabolism take place inside cells continuously and concurrently, so at the same time. So if we look at the role of ATP in metabolism, so ATP can store energy in this high energy. So if we look at the role of ATP in metabolism, now ATP temporarily stores energy in its last sort of high energy phosphate bond. When water is added and the phosphate breaks free, that releases energy to do cellular work or processes. The ADP then and the phosphate can sort of be used then to resynthesize ATP, and this involves energy input from nutrient catabolism. Okay, so we're going to move on to look at carbohydrates and be able to state the various sort of dietary sources of carbohydrates. Polysaccharides are complex carbohydrates that need to be broken down into simpler carbohydrates before they are absorbed. Polysaccharides are found in vegetables and grains. Starches are found in things like bread and, pasta and things. And cellulose is a carbohydrate that's a component of most plant cells, and it passes through the digestive system without being broken down. But it does still have an important role to play in the digestive system because it adds bulk to feces and kind of keeps the whole digestive process moving. Disaccharides are found in refined sugar and dairy products, and they also need to be broken down into monosaccharides before they can be reabsorbed. And monosaccharides are found in fruits. These are the ones, that can be directly absorbed. Glucose is the carbohydrate most useful to the human cell. And other monosaccharides such as fructose and galactose can be converted into glucose in the liver. So I'm just going to give you an overview now of glucose metabolism and the generation of ATP. We're not going to go into this in any detail, really. If you need to know this for your course, you'll do this in biochemistry subjects. So once glucose is taken into cells, it undergoes metabolism, and there's 3 steps involved. The first one is glycolysis, okay? It occurs in the cytoplasm of all human cells, and it occurs in both aerobic and anaerobic respiration, so with or without the presence of oxygen. 2 molecules of ATP are required with 1 glucose molecule to produce 2 molecules of pyruvate, and we're not going to talk about that too much now. And that releases 4 molecules of ATP. And this, yields then a net 2 ATP because you had to input 2 to get the 4 out. So you have a net yield of 2 ATPs. The second step then is the citric acid cycle, which is also known as the Krebs cycle. It occurs in the mitochondrial matrix of cells and only in aerobic conditions. Glucose itself can't enter the cycle, so it must be converted to pyruvic acid. So pyruvic acid from the glycolysis step enters the citric acid cycle. So what you end up with is this 2 pyruvic acids giving carbon dioxide plus acetyl coenzyme A plus 6 waters, plus all of these plus 2 ATPs. And that's what we're interested in here. So the net yield of ATP from the citric acid cycle is 2 ATPs. Okay, it's where it gets funky. Okay, the electron transport chain. It's a chain of molecules that are embedded in the inner membrane of the mitochondria. What we have are these high energy electrons and their protons that were removed during the citric acid cycle. So these ones here, they enter the membrane. And as electrons then move down the chain, they release small bursts of energy to pump the protons between the inner and the outer membranes of the mitochondrion. So this yields up to 34 ATP, right? So it's a very efficient way of getting, far more ATP than, say, what you would get in anaerobic respiration, because these ones only occur under aerobic conditions. So it does yield up to 34 molecules of ATP per 1 glucose molecule, right? That's the highest, but it's not always that. And so therefore there is a total sort of yield of 38 ATP molecules, 2 from here, 2 from here, and 34 from here for 1 glucose molecule. Okay, so if we look at a summary here of the energy extracted from glucose, here we have glycolysis. So we have glucose being converted to pyruvate. You need 2 ATP molecules to do that. And it produces 4 ATP molecules, but then the net ATP is 2. And then we're going to go through all of this here in the citric acid scale, or the Krebs cycle, and the electron transport chain. And in this cell, this is releasing 34 ATPs. And if you add on those two there, you get 36, okay? In some cells, that are highly metabolic, such as heart and liver cells, it can be up to 38, as I mentioned on the previous slide. Okay, so let's look at the difference between aerobic and anaerobic respiration. It's fairly straightforward. Aerobic, oxygen is present, glucose is fully metabolized to water and carbon dioxide. 38 molecules of ATP from each one glucose molecule, and those 3 steps that we summarized. Anaerobic respiration, there's no oxygen, so glucose molecules only go through glycolysis. Pyruvic acid molecules are converted to lactic acid, fermentation, instead of going through, the citric acid cycle, and only 2 molecules of ATP are released from that process. And the lactic acid then is released, and it's, transported back to the liver to be converted back to glucose for later use. We're now going to look at the difference between these terms: glycogenesis, glycogenolysis, and gluconeogenesis. In terms of, unused glucose, it needs to be stored. In the short term, glucose is stored as glycogen, and glycogen is a large branched molecule made up of chains of glucose molecules. This occurs mainly in the liver when it needs to be used by the whole body, and it can also occur in skeletal muscle. But the glycogen that's made in skeletal muscle is only used for muscles. So glycogenesis then is a series of chemical reactions in which glucose molecules are joined to form glycogen. Yeah? Glycogenolysis, so olysis meaning splitting, is the reversal of glycogenesis. So breaking down glycogen to release glucose. You might do that, when your blood glucose levels drop. Gluconeogenesis, so new glucose formation, is the creation of new glucose from glycerol or amino acids if glycogen stores run out, to form glucose. And this, of course, is also occurring in hepatocytes.